All right, the final part of this chapter is to discuss techniques used for investigating DNA and RNA. And so they take advantage of certain properties of these molecules. So one example is how they absorb UV light. One example is the fact that they can be denatured and renatured. So this means that you can separate the double-stranded DNA into single strands. An example of that technique is FISH, so fluorescent in situ hybridization, and then finally electrophoresis of nucleic acid. So these are four techniques that take advantage of fundamental processes of DNA or RNA. So the nucleic acids, so they absorb UV light strongly at the 254 to 260 nanomolar range, due to the interaction between UV light and ring systems of the bases. So the nucleobases, let's see if I can go back and find an example of a nucleobase. So the nucleobases almost always have this phenyl ring, and these rings, when they have these double-stranded, or these double bonds within them, they can actually absorb light and at a particular wavelength. So the energy of the photon has to match kind of the resonance of this, and if it does, then it's actually able to absorb this light and actually change kind of the orbits of the electrons within it. So it absorbs that energy and changes the energy. And so experimentalists were able to figure out that this could be a useful way to quantify how much DNA is in a sample. And so this is the absorption range. So if you looked at how much light that DNA absorbs, so the absorption right here is centered over the 260 nanomolar wavelength. And so if you shine light that's 280 or 290, the DNA will not be able to absorb that energy. If you send in 240, again, the DNA will not be able to absorb it. So you have to have very particular wavelengths of light that the DNA is able to absorb. And if it does, then the light will be absorbed. Proteins, on the other hand, their action spectrum is more along the lines of 280. And so typically the way you quantify the amount of DNA is you compare the amount of absorption at the DNA to the amount of absorption at the protein range. So if you look at the 260 and compare it over 280, and your sample is almost all DNA, then you expect to see a nice large ratio. So the ratio 260 over 280 is used to quantify how pure something is. So if you have a lot of protein, then you'd also have a lot of absorbance at this range as well. And then you would just simply use the 260 in order to quantify how much DNA is. So there's a relationship between the absorption and the amount of DNA within a sample. So these are called spectrometers, and you will find them throughout the lab. So if you ever do undergraduate research, for example, you might use a nanodrop or something like that, and that's the basis of that technology. This also explains why UV light is so damaging. So if you stay out in the sun for a long time, that can cause cancer, and that is because the sun releases UV waves. And so these UV waves in this range can be absorbed by the DNA within your skin, and that can actually lead to damage of DNA. So that energy initially gets absorbed into the electrons of the nucleobase, but eventually that energy has to go somewhere. And as a result, that energy can go in different ways. And one way is that it actually mutates the DNA. And so if that happens, then that can be inherited within additional cells. It can also break double-stranded DNA, and that can cause all sorts of issues for the cell that either might lead it to die, or potentially it can mutate it in such a way that these cells can become cancerous. So this is kind of the molecular region why UV light is dangerous. We also talked about the fact that you can denature and renature nucleic acids. So normally DNA is found in a double-stranded form, but what happens if you take this sample and you apply heat? So So if you heat this sample to something like 99 degrees, so before water boils, right before water boils, you've added a lot of heat to the water sample. And so that energy is actually able to overcome the hydrogen bonds. So the hydrogen bonds are initially keeping those strands together, but as the single strands get more energy from the higher temperature, they're eventually able to break themselves apart. And so why is this useful? This is useful because you can now add additional types of sample that is complementary to the DNA. So for example, if you added RNA that was complementary, so it had the complementary nucleotides in the right sequence, and then you lowered the temperature from melting, you now can create these hybrid forms, so complementary duplex of such as like DNA RNA hybrids. And so now you have a single strand of the original DNA, so this was unable to find a partner, and then the RNA was able to find the wild type complementary strand. So fish is a great example of how this can be useful. So it uses fluorescent probes to monitor hybridization. So cells are heated to denature DNA, fixed to slides, and then subjected to hybridization. And then single-stranded DNA is added, and these probes contain fluorophores that allow you to visualize where within the cell something might be. So you imagine this RNA molecule that originally is just an RNA molecule. Now you put on something that's bright red or bright green, something like that. So it's like some sort of fluorescent dye that will emit light. And so you do this, and now you look within the cell, and so you can see, for example, this is the location of one segment of the DNA within a bacterial cell. So we can see where within the cell this particular position of the genome is located. So this red just shows you like the entire bacterial cell. And then this is the probe. You have a bunch of probes that are fluorescent that are all going to the same place, which is just one position of the genome that you might choose. So you can use FISH to look at the locations of DNA within the cell, and you can also use FISH to look at the locations of RNA within the cell. So it's a very common process that's been used in laboratories, in lots of different species. When you look at multicellular organisms, it's a way to see where a gene is expressed. So like what sort of tissues is a gene expressed? Because these probes will only hybridize at a position where the mRNA is within the cell. All right, the final technique is electrophoresis. So electrophoresis allows us to separate things by size. So DNA sequencing, which we'll cover in a couple chapters. The first ways that we ever did DNA sequencing was taking advantage of the production of different sized DNA fragments, and then these can be resolved on a gel. And so you use an agarose gel. So agarose is a material from seaweed that you purify from seaweed is the typical source of it. But what it does is it produces this gel-like material, almost like Jell-O, but like a little harder. And if you run DNA through it, it separates them out by the size. And then you can actually visualize the DNA afterwards by using dyes that intercalculate in between nucleobases. So one of the things we found that's useful for these techniques is the idea of dyes. And again, we're gonna find dyes are very useful in all aspects of biology. All right, so let's talk a little bit about how this is done. And so you create an agarose gel, and this agarose gel has pores that these strands of DNA can move to. So you would load the DNA at the well. So this is where you'd actually put your sample initially, and then you would try to get it to run towards this size. And so you might think, how am I able to do that? Do I use gravity? No, you don't use gravity. What you do is you create an electric field. So you create an electrical field where there's a positive side and a negative side. So the cathode is here and the anode is here. So this takes advantage of the fundamental property of DNA that it is negatively charged. So again, because of the phosphorus atom is an acid, you have one less hydrogen as normal, and so as a result, the overall charge of the DNA molecule is negative. So it steals that electron from the hydrogen and releases the hydrogen, the positively charged hydrogen ion. And so now we have a force that's slowly causing the DNA to move through this gel, and the speed at which it happens is based upon the length. So the force is proportional to the number of nucleotides, but then there's a much less resistant force on going through the pores based upon the size. So these shorter fragments actually run faster than the longer fragments. And so if we do this, and let's say we had a sample that had 4 different size pieces of DNA, so you had millions of pieces of this fragment, millions of pieces of this fragment, millions of this, then after running this out on a gel for like 60 minutes, you'd be able to separate these bands. Now this is actually what something looks like within a laboratory. So this is an example of a gel box. So in this case, you would have the gel be right here, and you would typically load the DNA sample somewhere right here. So these would be all the different wells. And then this power supply right here is what's actually used to create the electric field. So the red indicates the positive, so this wire eventually moves here. And so this is where the positive charge is, this is where the negative charge, and that put the force on the DNA to run towards this way. You take this sample and this buffer, so this is liquid, and this buffer contains a dye, so that if you illuminate with UV, you're able to see the bands of DNA within it. So this is just an example of what one band would look like. This is kind of a smear of DNA, and then you also have samples where you have 4, 5, 6, etc. bands. So you can have multiple size bands within a population, or you can have a single band. And so again, we're going to see how this is used in future chapters in order to perform things like sequencing, restriction mapping, etc. All right, so that is the extent that we're going to cover chapter 10, and that is it.